அப்படியா? தெரியாம வந்துருச்சு சார். சாரி. கிளாஸ் ஆரம்பிச்சிடலாமா சார்? அதர் ஸ்டூடெண்ட்ஸ் ஆன்லைன்ல இருக்காங்களா? ஓகே 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 சார். ஆன்லைன்ல ஓகே. மைக் ஆன் வச்சுக்கணுமா? ஞாபகப்படுத்துங்க. ஓகே போகும்போது சார் மைக் வச்சுட்டு போகணும். Okay, we'll start. So what's heredity, Rohit? Heredity. Okay. How the characters are inherited. That's heredity. How the characters are inherited. Inheritance of traits. That's heredity. In many ways you can. How from parents to progeny. Characters are inherited. That process, that principle is heredity. Okay, fine, good. And there are the character do not inherit it as such. They follow certain rules for the inheritance of character from parents to progeny. There are certain rules. Okay, so that we are going to study. Rules of inheritance. Okay, mechanism. In simple what? Mechanism. How characters are inherited from parents to progeny. They have given you a task here. Okay. Observe the as of all your students in the class. Okay. Prepare a list of students having free and attached air lobes. Calculate how many percent of free. Look at that. This is free air lobes. Can you see? That's free. And this is attached. Free and attached. Two types of air lobes. Okay. When you calculate, then you can come to know that the numbers, basically the percentage, and what is dominant, in the sense either this is dominant in the sense in 40, if students are 40 in number, maybe 30 here or 10 here. If you go to the next class also, if you calculate, if their students are 60, you can see 15 and then 10. This ratio, some rules, okay, this free lobe inherited in certain ratio. Attached air lobe inherited in certain ratio. So like that, some little activity they have given. Means every character and its inheritance are based on certain genetic principles. Okay, heredity is based on certain rules and law. Okay, what you should understand, heredity is based on certain rules and laws. Okay, fine. How we came to know that? Because of Grahor-Mendel's experiments. The laws, how we came to know that? Grahor-Mendel's experiments. Okay, fine. Now, where the, of course, paternal, maternal means what? DNA. What is paternal DNA? What is maternal DNA? Father DNA. Paternal means what? Father DNA. Maternal means what? Mother's DNA. Okay. How characters are inherited from parents to progeny? From the paternal and maternal DNA that is inherited from the parent to progeny. Okay, in sexual reproduction. Can you understand? So DNA only, both. The characters are influenced by what? Every trait we get from, every trait, all our character we get from paternal and maternal DNA, from Appa and Amma. Can you understand? So that thing. Okay, fine. Now, in 1865, okay, who is the father of genetics? Okay, means characters are inherited from parents to progeny. Okay, inheritance of characters is because of some factors. He is the one who used a term called what? Factor. What is Mendelian genetics? Factors determine character. Okay, what is Mendelian genetics? Factors. You can't see? Oh. You can't see, are there? Can you see now? Ah. Oh, only 50% you saw. Okay, means factors determine characters. Okay, can't see, right? Factors determine characters. Can you see? Factors only determine characters. That's the final conclusion he gave. Okay, who is Grahor Mendel? He's a monk. Monk na Turavi. Okay, he's a monk. He lived in a monastery. and he went to study science and mathematics in University of Vienna. But teacher training, okay, father went for the teacher training course. Actually, he failed in his experiments. Okay, so but he did not receive a certificate, teacher certificate. He did not receive. Okay, so he went back to his monastery, and there a little god, church, monastery means church இருக்கும். Church பக்கத்துல இருக்கிற ஒரு சின்ன garden, this much of space, very little garden, church. இப்படிதான் church இருக்கு அப்படின்னு சொன்னாக்கா, and this is the garden, Mendel's garden. Okay, here only he carried out his research. Okay, fine. And for his study, pea plant. What is the scientific name of pea plant? Pea plant, patani chedi. What is the scientific name? Pisum. Have you heard about this? Sativum. No, no. Pisum sativum. Okay, Pisum sativum is the plant. What he carried out, with that only he carried out this research. Patani chedi nu solluvom. Scientific name. All the scientific names are Latin based only. Homo sapiens, scientific name Latin. The scientific names: bacteria, Escherichia coli, bacteria name. Mangifera indica, scientific name of mango, Mangifera indica. Okay, Oryza sativa, scientific name of rice. Can you understand? So all scientific names, okay, binomial nomenclature introduced by Carolus Linnaeus. Okay, all are scientific name. So Pisum sativum is the scientific name. Common name is pea plant. Regional name, நம்ம ஊர்ல அதுக்கு பேர் என்ன? பட்டாணி செடி. Is it clear? And blending experiments. What is called blending experiments? Hybridization experiments. Blending experiments means what? Hybridization. Hybridization. What is the layman term for hybridization? Crossing. Crossing of parents. Simple. That is called what? Hybridization. What is the plant? Pisum sativum. Pea plant. Okay. Is it clear? And with his experiments on pea plants, he helped him to arrive the laws of inheritance. Okay, helped him to arrive what? The laws of inheritance. Okay, fine. Now what you should know is some questions they may ask, like what are the 7 characters selected by Graham Mendel? Okay, what is the question? 7 characters selected by Graham Mendel. You know that? 7 characters selected by in the pea plant. 7 contrasting characters he selected. Okay. You have that yellow material? No, just check the 7 characters selected by Graham Mendel. Okay, I think page number, oh, not 27, page number 27. Okay, yeah, stem height, flower color, flower position, pod shape, pod color, seed shape, seed color. Okay, basically they are the 7 characters selected by Mendel. Okay, like round or wrinkled, you can see something here, round or wrinkled, tall or short, white or violet, and like that. Okay, fine. So please know this table. Okay, fine. And before that, some terms you should know. Okay, you should understand some terms. What are the terms? The reason for choosing pea plant. It is not a seasonal plant. In the sense, in all the seasons, the plant can be produced. Okay, in all the plant, it can be produced, and easy cultivation. Okay, easier it can be cultivated, and then both self-pollination and cross-pollination are possible. Okay, both self and cross-pollination are possible. In the sense, it is a bisexual plant. What is called bisexual plant? Male and female components, androecium, gynoecium, are present in the same plant. Okay, fine. And then short life cycle. In few weeks, you can see from the seeds, the whole plant developed in few weeks. Okay, and in Kutti lab, see, two folds than this classroom. In little classroom means the space is lesser than the center, very less place, and it can be cultivated. And the 7 characters which selected by Mendel are observable, visible, easy, phenotypic traits. All are phenotypic traits only. Okay, you can see, and no other variations. Can you understand? So like that, there are many reasons for which Mendel selected this plant. Can you list? Tell me. Can you list? What are the things? Look at that. Well-defined character in the book. Take it. Well-defined character means 7 contrasting characters, and then it is bisexual flower. Can you see that? It is there. It is a bisexual flower, and then both predominantly self-pollination, but you can do by emasculation and bagging. Yeah, down you can see some technique, emasculation and bagging. So means it is possible to do cross-pollination, both. Okay, and then easy hybridization. You can crossing experiments are easy. Okay, that's all. So these are all the main reasons. Okay, he selected the plant, had more advantages. Okay, fine. And moreover, he failed in his teacher training exam. He wanted to do research on animals, but the monastery did not allow, since hybridization, breeding in animals, cattle, he wanted to do. But they said, you are a monk, priest. Animals involved in sex. Reproduction is a sexual process. If you read that, your mind gets what? Disturbed. Okay, that's why they didn't allow. But Mendel thought, even plants, or can you do it in plants? He asked. Yes, you do it in plants. Then he thought, even plants can undergo sexual reproduction. Here also male-female plant is there. All these things. But why should I explain all these things? Let me do it in plants. He also did lot of animal experiments also, but he didn't include like in all his research. Just this part he published. Can you understand? Almost for 7 years he worked. How many years? For 7 years he worked. More than around 2,500 plants. Like he was involving more than 2,500 plants. Okay, number of 2,500 plants he used actually. Okay, finally, but he documented everything, and he is the one who introduced statistics into science. That experiment, statistical values, mathematical representation. But since he got mastered in that, when you give 2 plants, he will say, if I cross this plant, 2,730 seeds will come. If you count that 2,732 seeds, and other 2,500 will be a round seed, and rest 200 will be a wrinkled seed. So like that, if you give 2 plants, one tall, one short, he will say, if I cross these 2 plants, and 7,000 plant will come. In that 6,000 will be tall and 1,000 will be short. So like that, from the plant and its seeds, when he observes traits, he can predict, he predicted. So that's how, like, the law, because he understood the law. If I cross these two, what will be the results? So that's the main thing about Mendel's success. Introduced mathematics and statistics into science, biology, and recorded all the things which he observed. Those are all the reasons he is successful in his experiments. Okay, fine. Good. And so before getting into that, you should be thorough with what some terms. What are the terms? Look at that here. Okay. And in that material, can you see when Mendel published his results? 1866, he published his results. Okay. But nobody can understand. Okay. But he recognized in 1900s only, means, but by the time he died. Okay. But Mendel did not know that he is the father of genetics. Nobody understood. But he said, my time will come. Because he knows, he discovered the principles of genetics. But nobody understood. But he said, my time will come. After 35 years, he died. After 35 years only, it happened. Okay. He published in 65 and 1900s only it was rediscovered. 3 different people, independent researchers. Who are they? The 3 independent researchers. Hugo de Vries, Karl Correns, and Eric van Teschmark. Okay, Austrian botanist. Can you understand? So they only like rediscovered Mendelian principles. Okay, and then fine. So what are the characters, terms he used? Dominant trait. What is called dominant trait? The trait appears in first generation. Filial generation. Can you see F1 present? Means, see, look at that. One tall plant. Okay. See, he crossed. This is one parent, tall. This is one short. Okay, they are what? Parents. Okay. The first generation will come. That is filial 1, F1 progeny. Filial 1 generation. First generation. Okay. Maybe all were tall. So, and tall only, but heterozygous for tall. And then in the F2 generation, he gets what? Look at that. In F1 generation, F1, he crossed one tall plant and stem, short plant. In the F1 generation, what is there? Only, he has what? Only tall. In the first generation, right? What is not there? Dwarf is not there. And then he did selfing. F1 with F1. What he observed? Like, look at that, tall. What he observed? 3 tall, 1 dwarf. This ratio is 3:1. Dwarf is reappeared. This is a simple experiments. We'll do all these things in detail, don't worry. Okay. So F1 generation, parents are crossed. The first generation is F1, and then the F1 is crossed. Second generation is F2. Filial. See, Filial. Filial generation. Or you can say first generation. First generation, F1 generation, filial 2, second generation. Okay, fine. So what is dominant trait? Look at that, tall one character, trait, short one trait. Trait means character. Okay, fine. What is there in the first F1 generation? What we see? Tall or dwarf? Tall. Then if 2 characters are there, the one which appears in the F1 generation is dominant. Okay. Similarly, like that, the one which appears only in the second generation, which is recessive. Okay. Look at that. What is dominant? You read the definition. F1 generation. Around the trait which appears in F1 generation is called dominant trait. denoted by capital T, capital T. Homozygous dominant. And then recessive trait means the trait which does not appear in F1 called recessive trait, but it appears in the second generation only, represented by small t, dwarf. Okay, fine. And then 4 type of crosses: monohybrid cross, dihybrid cross, test cross, back cross, reciprocal cross. 5 type of crosses. Okay, what are they? Monohybrid, dihybrid, trihybrid, back cross, test cross, reciprocal cross. Okay, what does it mean? Monohybrid means what? Monohybrid, study of inheritance of one pair of contrasting character. Study of inheritance of one pair of contrasting character. That is, if for example, look at here, one pair of contrasting character. This is parent is homozygous for tall. Another one is homozygous for recessive. One pair of character. How this pair is inherited? That is monohybrid cross. What is dihybrid cross? Look at that. Inheritance of 2 pairs of contrasting character. Okay. For example, we will be studying or y. Seed, 2 characters in the sense. Look at that. Seed shape is round. Seed color is yellow. Another one is, this represents what? Seed shape is green, means wrinkled, yellow. Seed color is yellow. That you can see in the next table. Can you see the table? Seed, pot shape. What is it? Seed shape. Look at that last 2. Round, wrinkled. Round is dominant, wrinkled is recessive. Like that, seed color, yellow is dominant, green is recessive. Okay, so trihybrid cross in the sense what? This is monohybrid cross, this is dihybrid cross. 3, for example, or you can say, means this is round, yellow, round seed, and then look at that, round seed. Okay, second character is what? Color which is yellow. Okay, third character what? The plant may be tall stem. Can you see 3 characters together? Another one may be, what is the other one? Short plant. Okay, and then the color is green. And then which is wrinkled, not round, just wrinkled. Green and then wrinkled seed, not the round seed. Okay. Okay, can you see? This is what? Trihybrid cross. Monohybrid cross, dihybrid cross, trihybrid cross. Okay. What is back cross? Listen. Here, they are the parents, right? These 2 are the parents. What are the parents? TT are the parent. What are the progeny? Correct? How do you describe this? Homozygous, tall, dominant. This one, how do you define? Homozygous means both are same allele. Homozygous. Short, recessive. Tell me, first one is, how do you full homozygous? Why homozygous? Homozygous means what? Both the genes are of same type. Both the alleles are of same type. It is homozygous. It is dominant. Heterozygous, look at that. One tall, one dwarf. 2 genes. Actually, one tall. But anyway, what is expressed there? Tall only will be expressed. Okay, so the description you should know. So, this is one parent. This is one parent. What you got? F1 generation. This is monohybrid cross. Now, Dihybrid, trihybrid cross. No. What is back cross in the sense? So this is F1 progeny, right? If F1 progeny, it is heterozygous into any of the parent. What is the parent? This is parent 1. This cross is what? Back cross. See, progeny with any one of the parent. Progeny with any one of the parent. I gave you one option. This progeny with this. What is the other option, Rohit? In back cross, what is the other option? One option I gave you, progeny, F1 progeny with parent 1. What is the other option? Parent 2. This is F1. Parent 2. What is the no progeny? What is the parental character? You can't understand. See, please listen. I'm telling you, this is one parent. Parent 1. What is the sex now? Male. This symbol represents what? Male parent. Another parent is okay. Both of the parents are crossed. This T and this T, this T and this T. Now, progeny will be like this. Yes or no? Here one gamete comes, fuses with this gamete. Here this gamete, this fuses with this gamete. They are called what? F1. P1, P2. This is F1, first generation. This is monohybrid cross. Correct? What is back cross? I'm giving you one option. F1. What is F1 here? This is F1 into parent 1. What is parent 1? This is one parent, right? This is crossed with this parent. That is one option. Now I'm asking you, what is the second option in F1? Means back cross. This with this one. Can you understand? How do you write this? T into okay, TT into, just tell me, TT into recessive T. So this is another option. This is called what actually? Back cross. F1 with any of the parent. Parent progeny ஒரு F1 ஓட F1 progeny இதோ ஒரு parent கூட cross பண்றோம். அதை என்னன்னு சொல்றோம் நம்ம? Back cross அப்படின்னு சொல்றோம். ஹியூமன்ஸ்ல பண்ண முடியுமா? முடியாது. இல்லையா? பண்றது இல்ல நம்ம. நம்ம அப்பா அம்மா கூட போய் சேருவோமா? முடியாது. பட் பிளான்ட்ல உண்டு. அப்படி பண்றதுக்கு பேரு back cross. ஓகே? monohybrid cross புரிஞ்சுச்சா? dihybrid cross புரிஞ்சுச்சா? trihybrid cross புரிஞ்சுச்சா? back cross என்னன்னு புரிஞ்சுச்சா? இப்ப test cross. அடுத்த cross பேர் என்ன? test cross. Test cross means, look at that. நான் உங்ககிட்ட ஒரு unknown character கொடுக்கிறேன். unknown அப்படின்னு சொன்னா ஸ்டெம்மோட டைட்டில் இதை எடுத்துக்கிட்டாலும், this is one condition. Maybe this or maybe this. Homozygous for tall, homozygous for heterozygous for tall, or homozygous for short. 3 கேரக்டர் இருக்கா? நான் உங்ககிட்ட கொடுக்குற ஒரு பிளான்ட் 3 கேரக்டர் அதுல எது வேணாலும் இருக்கலாம். இது நீங்க இது என்னன்னு கண்டுபிடிக்கிறதுக்கு என்ன பண்ணோம்? What you have to do? Cross the unknown versus known recessive. Test cross அப்படின்னா என்ன? Unknown versus known recessive. அதுக்கு பேர் என்ன? Test cross. டெஸ்ட் பண்றது, கொடுத்திருக்கிறது என்னன்னு சொல்லிட்டு பண்றது பேர் என்ன? Test cross. ஓகே, ஃபார் எக்ஸாம்பிள் இப்போ TT இருக்கு, இதான் நான் எடுத்துக்க போற பிளான்ட். இன்ட்டு TT பண்ணலாம். ஆர் செகண்ட் என்ன? TT இன்ட்டு T T பண்ணலாம். Third என்ன? T into. Can you understand? So in three way, can you do that? See, test cross na enna solunga paakalam. Can you define? Unknown, which is given, versus known recessive, not a dominant one. Known recessive. That is what? Known recessive. What is the character? How do you write? TT. So this is test cross. Okay. Please understand these things. What are the things you saw now? Monohybrid cross, dihybrid cross, trihybrid cross, back cross, test cross. Now, reciprocal cross. What is the next cross? Reciprocal, changing the sexes. For example, Kaushik, are you following? Kaushik? Okay, this is male plant. Okay, this is what? Male plant. Character is T, tall, dominant. Into female plant. What is the female plant? Homozygous for dwarf. This is a symbol you give for male and female, right? And contrasting to that, reciprocal cross means the male plant will be this, and female plant will be what? Can you understand? Inga parpa, tall plant female, first cross la, cross 1, cross 2. First cross la yaru tall plant? Male is tall, female is short. Second cross la female is tall, male is short. அப்ப இதுக்கு பேர் என்ன? Opposite, vice versa பண்றீங்க. இதுக்கு பேர் என்ன? Reciprocal cross. Can you understand? Fine. Now, the other terms we should see. What is genotype? What is phenotype? இங்க பாருங்க. இப்ப நம்ம யூஸ் பண்றோம்ல இந்த terms, genetic composition. இதுதான் என்னன்னு சொல்லுவோம்? Genotype. Genetic composition is called what? Genotype. என்ன gene character determine பண்றதுன்னு சொல்லிட்டு. Phenotype na enna? இந்த plant எப்படி இருக்கும் பார்க்கிறதுக்கு? How it looks? Tall. Phenotype. பார்க்கிற character. நம்ம visible character is called what? Phenotype. What is genotype? Genetic composition. Phenotype, visible, what we see. What determines is genetic. What you see is phenotype. Can you understand? Phenotype. See, because to understand genetics, you should understand lot of terms. Okay? So genotype, phenotype. And then what is gene, allele, multiple allele? Okay? Have you heard about this term? Gene, allele, multiple allele. Gene, allele, multiple allele. What is gene? Factor which determine character. Is what? Gene. நம்மளுடைய character determine பண்ற DNA ஓட ஒரு பார்ட் தான் என்ன? Gene. Okay, நம்மளுடைய characters எல்லாமே determined by the factor which determines the character is gene. And the gene ஓட alternate form. Okay, alternate form. They call it as what? Alternate form of a gene. What is the alternate form of a gene? Alternate form. For example, in Mendel's experiment, one gene gave round color, round shape. Another gene gave wrinkled shape. Round shape, alternate form is wrinkled shape. So the gene which gave round is one allele, the gene which gave wrinkled is another allele. So alternate form of a gene. Can you understand? What is a gene? segment of DNA which codes for a character. Allele means allele, alternate form of a gene. Alternate form, another form of a gene. One gene gives black color eye, another gene gives brown color eye. One gene gives straight hair, another gene gives curling hair. One gene gives fairness, one gene gives darkness. Okay, means alternate form of a gene. So what is called multiple allele? To determine one character, you have many genes. More than 2 genes determines one character. Multiple allele. For one character, many allele. Many allele. Multiple allele means what? Many allele. Many now 2 plus allele. 2 and more. More than 2 gene for one character. For example, humans love blood grouping. What is it? Blood grouping. How many blood groups we have? A, B, O, AB. Okay, 4 groups. There are 2 variations, positive, negative. A positive, B positive, O positive, AB positive. A negative, B negative, AB negative, O negative. Yeah, A1, that's little variation, just one protein variation. But see, more than 200 distinct blood groups are there. More than 200. But A only, in A1, chinna protein change is there, but that doesn't matter. Okay, A, B, O. They are the major blood groups, right? A, B, O. This is determined by how many genes? 3 genes. 3 alleles determines one character. Blood grouping is one character. Determined by how many alleles? 3 alleles. Can you understand? So that concept is called what? Multiple alleles. Can you understand? That concept is called what? Multiple alleles. Okay. And you know what is homozygous and heterozygous condition? What is homozygous condition? Same type of genes. For example, from Appa also you got T, from Amma also you got T. In diploid organism, 2 alleles. For every character we have 2 alleles. For every character we have 2 alleles. From Appa also we got tall gene, Amma also we get tall gene. So both are same. Appa, this is homozygous. Okay, and what is this then? Appa tall, Amma short. Heterozygous. Heterozygous. Hetero means different alleles. Same allele, homozygous. Different allele, heterozygous. And what is this? What is this? Small t, small t. Homozygous? This is homozygous or heterozygous? Homozygous. Dominant or recessive? Recessive. Very good. So this is homozygous dominant. This is recessive. Recessive dominant. This is heterozygous. Can you understand? So these are all the terms you should understand. So please go through the terms. What are the terms we discussed? Dominant triad, recessive triad, monohybrid cross, dihybrid cross, trihybrid cross, reciprocal cross, back cross, test cross. Homozygous condition, heterozygous. Genotype, phenotype. Gene, allele, multiple allele. So go through that terms. If you understand this only, and then rest you can understand. Which one? Dwarf, short. Dwarf means short. Dwarfism. What is gigantism? Dwarfism. Gigantism, பயங்கர ஒரு பெரிய சைஸ் வருது, giant. Dwarfism, shortness. So list down the terms. What are the terms we discussed? dominant, recessive. Hmm. 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 Gene allele, multiple allele. Hmm. Very good. All these definitions you should know. Meaning and definitions you should know. Okay. And then what else we discussed today? Name of the plant is pea plant. Scientific name is Pisum sativa. Okay, yeah, fine. Why Governmental selected Pisum sativum? We saw no. Why Mendel selected that plant? Hmm. Hmm. It's a bisexual flower. Easy self-pollination, easy cross-pollination, emasculation and bagging can be done. All interesting characters, 7 contrasting characters. Characters are easily observed. Yeah. So that's why we selected, Grafar Mendel selected that plant, pea plant. Who rediscovered Mendel's work? Hmm. Ah. Hugo de Vries, Dutch person. Karl Correns, Van Erich von Teschmark, Austria. Mendel also from Austria. Okay, an Austrian monk. Austria is a little country. Austrian monk. Can you understand? So all these people. Okay, this is what we see. What are the 7 contrasting characters selected by Mendel? Hmm. Hmm. Read that. That's 7 chart. 7. Read it and just make the chart and show me. Without seeing that, you should list able to tell or write. Kaushik, are you there in online? No. Yeah, you can tell even. Okay, like this first. What are the 7 characters? Stem height. Hmm. Very good. Okay, now dominant right and recessive right. What is, you know, what are they? What is pod? Hmm? Pod means what? Pod color, pod shape. What is pod? பீன்ஸ் எல்லாம் பார்த்திருக்கீங்களா? பட்டாணி செடி உள்ள இருக்கிறது சீடு, மேல அதை கவர் பண்ணி இருக்கிறத என்னன்னு சொல்லுவோம்? பாட். ஓகேயா? அதை சுத்தி கவர் பண்ணி இருக்கிற அந்த டிஷ்யூ தான், அதுதான் என்ன, அந்த மூடி தான் என்ன? பாட். அவரைக்காய் பீன்ஸ் எல்லாம் பாக்குறீங்களா? மேல இருக்கிறது பாட், உள்ள இருக்கிறது சீட். ஓகே, ஃபைன். நவ டெல் மீ தி வேரியேஷன். Stem height, actually tall is dominant, dwarf is recessive. அப்புறம் no, purple is dominant. Flower position, axial and terminal. What is axial? See, a plant is there, in along all the axis, if flowering is there, then that is called what? Axial. Okay, terminal in the sense, only in the edge you can see, in the terminal end you can see the flower end. That is called what? Terminal. This is terminal, this is axial. Along all the axis. Can you understand? Flower position along all the sides or only on top? Fine. Next, look into the image. Can you see? This is what type of plant? Axial or terminal? All are axial. Can you see? Along the all axis you can see the flower. Okay, that's why it is axial. If flower only present in the end, then it is terminal. Okay, fine. What is full and constricted? Pod shape, full constricted, right? Full inflated. What are the two characters? Full and inflated. Full means what? This is the pod. This is full. Inside that what you have? Seed. This is full. Constricted in the sense, puriliya. Okay. Can you see? This is full. Constricted means what? Can you see here? You have the seeds. Inflated or constricted. What is the difference between these two? Here it is full. அப்படியே முழுக்கட்டியா இருக்கு. Okay, but இங்க பாத்தீங்கன்னா inflated or you can say constricted. Okay, so all these 7 round, wrinkled, you know, round, wrinkled. Okay, yellow green color you don't have to worry about. So all these things are the important characters. Okay, what is emasculation? Masculation, removal of anther from a flower to avoid self-pollination. You should remove the anther. That is emasculation. What is bagging? Cover the flower or the stigma with what? A bag, plastic bag or whatever. Cover the flower, protecting cross-pollination. Preventing self-pollination is emasculation. Preventing cross-pollination is covering. Stigma is bagging. Bagging, just bagging. Emasculation, bagging. Okay, so these are all the terms which helps you to understand genetics. And tomorrow we will see about how Gregor Mendel carried out his monohybrid toss and the loss, dihybrid toss and the loss. Okay, take care. Thank you. Boring, yeah? Alone. Please know about these terms. Okay, read that yellow book.